Hello, Eastview scholars, and welcome to this edition of Sharp Shorts, where we talk genetics, and more specifically, as we roll into this part of AP biology, we're going to first just look at Mendel and how he came up with his theories of how traits and genes are transferred. Remember, in Mendel's time, he had no ability, no concept, no understanding of cellular parts. The technology did not allow him to know anything other than maybe things were made up of cells, so he called these things heritable characteristics. And the competing thoughts at the time were that you could have two methods of inheritance. One, the blending model, is that whatever genetic heritable traits you got from your parents were just you were a blend of them. And you'll say, well, but, then how could that happen? And stuff with traits that might skip a generation, etc., etc. Well, there was just the same questions with Mendel and those who believed in the particulate theory that you get discrete, which we now know are genes that are on the chromosomes from your parents. And when we look at in future videos things like incomplete dominance, it supported blending also in a theory. So those were the two battling theories, and Mendel, if you didn't know, Gregor Mendel was a monk, and he chose pea plants because they had some distinct characteristics that you could trait in plants. You could get some decent quick generations. Well, he had plenty of years in the monastery to do things, but he was able to grow these peas and look at the different characteristics and come up with his theories. Very much like John Dalton in his first theory of the atom, without being able to see the atom or have the technology to narrow down anything about the atom, he did a pretty good job of coming up with a method of the atom, as Mendel did for a method of transmission of what he called heritable traits. Anyhow, here's what Mendel's belief was in the particulate model, that you could have alternate versions of genes accounting for the variations in inherited characteristics. You get at least two copies of the gene. If the two versions that you get, if the two alleles at a locus differ, then one determines the appearance, one is dominant over the other, and they must segregate during gamete formation. Okay, now that's kind of telling you what we're going to find at the end. Let's make sure that we are sure of our terminology. The allele, as we now know it, the allele is that spot on the chromosome where that gene is. The locus is the position of that gene. Here's a purple, here's a white, so you had two different variations of that gene on your homologous pair of chromosomes. Character is the flower color. Traits are they purple or white? Now, if you get both of the alleles for the purple flower, then you are homozygous dominant. If you get one of each type, then you're heterozygous, and this will make you a carrier, even though the white gene here isn't expressed and the purple one is. And then if you get both the alleles for the white flowers, lowercase p and h case, then you're homozygous recessive. And part of the question we have is, how do we know? I'm telling you purple is dominant over white. How do you know that? And what comes up later in the blending theory is maybe the heterozygous isn't one or the other, but that comes later, which supported those who believed in blending. Anyhow, that's our terminology. We have homologous chromosomes here. In other words, that character, you got one from dad, one from mom. The locus is the position of what we now know as a gene, or that heritable trait, and the allele is the trait that you have. Okay? So, in setting up as an experiment, and oftentimes that is the key to being successful, Mendel chose to track those characters that really, in all the growth and breeding of the plants, had two alternative forms. And he also made sure to self-pollinate so that he had true breeding, which really just means he either had homozygous dominant or homozygous recessive. That's the only trait they had. You can do that by self-pollinating those and essentially working out the heterozygotes and stuff so that every time you breed you're only getting all purple or all white. Okay? So, in his experiment, what he would do was blend, hybridize a true breeding purple, a true breeding white, or whatever the traits were, seed color, wrinkled or round seed or whatever, but he would only put in what's called the P generation, the parent generation, the opposing homozygous, dominant, and recessive, and breed those together. Okay. Then your first offspring is the F1 generation, and then you cross those F1 individuals, filial 1, and you produce an F2 generation. And part of the reason for breeding multiple generations, at least going to the F2 generation, if not beyond for Mendel, is to answer that question of traits that might skip a generation and solidify the theory that the traits and everything about you are those heritable characteristics that are passed down from your parent, of which you may not see them in a generation, but you might be a carrier. Okay? So, here's where he supports his law of segregation. Crossed true breeding white and purple flowers. All the F1 hybrids were purple. You'll see that picture in a minute. When he crossed all the F1, some were purple, some were white, and it was in a 3-to-1 ratio. Okay, so here we go. Here's our P generation. These are true breeding homozygous dominant purple. These are true breeding homozygous recessive white. First generation was all purple flowers. Okay. The second generation, though, had a 3-to-1 ratio of purple to white. Okay. Mendel's belief was that only the purple flower was affecting the color in that trait. That allele was affecting the F1 hybrids. And so that's why he called purple dominant to white. The factor for white flowers was not diluted, as blending would say, or destroyed, because it then popped back in the F2 generation. Okay. And Mendel observed the same exact pattern in 6 other pea plant traits. As you can see, flower position, seed color, seed shape, pod color, and stem length, all with true breeding plants going through the F2 generation, all had that 3-to-1 ratio, as you can see by his numbers. Okay. And that's what he called heritable characteristics that we now know are the genes and the different allele of those traits. Okay. 3-to-1 inheritance. And here's the 4 concepts we started at the beginning, which we're going to wrap up and discuss. First, alternative versions of genes account for variations in characters. For example, the gene for flower color exists in purple and white. These alternative versions are now called alleles, and each gene resides at a specific locus on a specific chromosome. Now, some of this is add-in of what we know now, but understand that it's all of its basis of alternative versions and heritable characteristics came from Mendel's experiments on pea plants. Second, each character in an organism inherits 2 alleles from one of each parent. Mendel made this deduction without any knowledge of chromosomes and how they operate or the role of it. Okay, the 2 alleles may be identical, as in the homozygous dominant, homozygous recessive of the true breeding plants, or they could differ, as in the F1 hybrids and heterozygotes. Okay. Third, 2 alleles differ, then one is dominant and the other is recessive and has no noticeable effect, and we saw that with the purple flowers. Okay, now we know that that's not quite true because of incomplete dominance and some non-Mendelian characteristics, but I think the thing to really understand about Mendel is he laid the foundation, and the numbers were true and they hold true. And then you can go off of his Mendelian genetics to understand alternatives or changes or differentiation in Mendelian genetics. Okay? And fourth, now known as the law of segregation and meiosis, which connects Mendel and his theories to how the inheritance of chromosomes, which we'll talk about at a later date. But in the law of segregation, the two alleles separate during gamete formation and end up in different gametes. Okay? Therefore, your egg or your sperm gets only one copy of the two alleles. Okay? And this allows for variation in distribution of the chromosomes to different gametes during meiosis, and allows for variation. And why, especially if you look at animals and humans and stuff like that, you do not look exactly like your parents. Either one or the other. You may have similarities, but you have that segregation. Okay? Mendel's segregation model accounted for that 3-to-1 ratio in the F2 generation. Possible combination sperm and egg can also be shown using a Punnett square for predicting, and that's when we went back and showed you homozygous dominant, recessive, and heterozygous for the flower colors. You always use a capital letter for the dominant allele, and lowercase represents recessive. And here's just a quick example with a blue beetle and a green beetle. Now this happens to be a heterozygous and a homozygous. These are not two true breeding. This one's true breeding. But you can see what you get here in the first generation is you get 2 out of 4 half become heterozygous, as one of those alleles went to one sperm, one went to another, one went to an egg, and obviously in a homozygous trait, then the egg is going to get the same thing. But then in a Punnett square for that trait, you can see how you can cross. You get half blue and half green with a heterozygous, homozygous trait. Okay. Genotype, we should say this. Genotype is what your actual traits are, the alleles you carry. Phenotype is what you see. All right. The allele expressed in the heterozygote is dominant. Okay. So, and again, this is just a retake of Mendel's flower colors. Okay. And now I'll ask you, as we do this, how do you tell which is purple or white, and how do you know that? Show the F1. So actually I would do a couple things. Do a true breeding Punnett square for purple and white, knowing that these are both homozygous, one dominant, one recessive. Show what the F1s are, then show the cross of the F1 generations to show the 3-to-1. Okay? And I think that would be great to figure out the genotypic and phenotypic ratio. Right? I'll have that. Our next video will continue with Mendelian characteristics and Punnett squares, but until then, Scholar on, Eastview.